Does the mic work okay? Okay. All right, so last talk we talked about toxicity, or I talked about toxicity, you had to listen. And this talk is going to be about how we try and get the hazard out of the molecule. So what can we do with it? Okay, outline green chemistry principle 4. So chemical products should be designed to affect their desired function while minimizing their toxicity. So what we want, molecules that are functional, because if we don't keep the functionality piece, the molecule is not going to have any impact. None of us would buy shampoo or whatever if it didn't work. So the molecule has to work for its intended commercial function, and we also want it to not be toxic. Learning objectives. I want to, in particular, focus on the toolbox. I'm going to characterize, to the extent I can in 20 minutes, the types of tools we currently have available and set the stage for the type of quantum leaps I think we need in that toolbox to help chemists design effectively. So this talk is focusing on mechanistic toxicology again. Keep going back to that. Stereochemistry is critical. And using that knowledge to design preferred chemicals and products. Historically, designing chemicals for function has not routinely incorporated safety as a design criteria. And that's a really big shift that needs to happen. We need to make sure that safety is considered. Again, this linkage between structure and function and toxicity. Predicting the linkage is where we really need to get to. So, and it varies depending on what industrial group you're in, when you need to know the toxicity implications, and so they don't, the toxicity, for example, doesn't become locked into your molecule. There's a certain point at which you can't necessarily make a change to a molecule. All right, the challenges here. These are really substantial. 500 to 1,000 or more new product chemicals developed a year. So think again about that process I described for pharmaceuticals. Could we undertake that process for 500 to 1,000 chemicals a year? It just can't happen. We'd go broke. And they're across a range of industries and chemical classes. So it's not like all these molecules would drop into one QSAR training set and that would give you an ability to predict toxicology. We need something better there. So the aspirations here are really to be able to build something into the R&D process and have safer molecules. And that can be done with chemical architecture. There are also other avenues open. For example, using different formulations that minimize the amount of chemical used, or by targeting a location within the body or somewhere else. So there are other tricks that can be used to minimize the quantity. And you know from what we've already talked about that using less is a good way to reduce hazard and risk, reduce risk. Sorry. As I indicated, there are commercial challenges. Some companies and some industries have only a limited amount of time before a molecule becomes locked in terms of its structure, and you can't really make changes. So let's talk about the hard work of predicting safety. There are some molecules that are very close structurally. So look at S-thalidomide, a teratogen, R-thalidomide, a sedative. Very close. Can we tease apart what it is that's specific about that molecule in a toxicological setting that we can be predictive in the way we look at new molecules? So very tough challenges here. Traditionally, this is the way compounds are looked at, structure activity, rules of thumb. Oh, it looks has some chlorine on it, may not be very biodegradable. QSAR, short-term assays, and then animal testing. The existing tools that are out there, so estimation methods, rules of thumb, the pharmaceutical industry uses a lot of in silico techniques to try and weed out toxicity in a hurry. Because if the compound's going to be toxic, the thing you want to do in that pharmaceutical industry is kill that compound early before you put a lot more investment in it. Toxicity to environmental species, there are models out there. This EcoSAR, EPA's EpiSuite. There are databases that are out there that have tremendous amounts of data that you can go and take a look at. Now what I want to do is, these are slides that a colleague of mine, Steve DeVito, provided. Steve DeVito is a good name, along with Adelina Vuchkova and some other people that are very active in this field, and I think they'll both be at the conference tomorrow. So designing molecules have low bioavailability, structural features, you pull those out. So there are known toxicophores. You can say these functionality aspects of a molecule are likely to cause toxicity. So you can try and minimize the usage of those. A continuation, so more toxicophores that are found in chemicals. So not using these is one way to minimize toxicity, minimize hazard in a compound. The biochemical basis, again, that mechanistic piece, knowing that can empower you to make good decisions about the molecules you're working on as a design chemist. Isosters are substances that have similar molecular electronic characteristics that can be slid into a molecule and very often maintain functionality, but give you the opportunity to have a different toxicology profile. Here's some more examples. Molecule on the left, too toxic to market. The molecule on the right is marketed as Tagamet. Another example, pesticides. You see the difference between the LC50, molecule on the left, molecule on the right. The reference dose is the daily exposure to the human population that's likely to be without an appreciable risk. Again, you see the differentiation between the two molecules based on that nitrogen. Another example, so the upper molecule, it's a known human carcinogen. The molecule on the lower right, much less mutagenic, but it's not clear yet whether it's carcinogenic. So these are some of the tricks that can be used. So a comprehensive across molecular class tool is what we're really hoping can be developed, and this is something that somebody wants a good project to work on. There's a lot of important work to be done in this area. Progress in toxicology has provided a lot of mechanistic knowledge recently, so there's a lot of causes for optimism in this. I'm going to point out this Lipinski Rule of 5 is a guideline that people have cited as an example of what would be useful in, for example, environmental toxicology and green chemistry to be able to provide some guidance. So a pharmaceutical has to bind with a receptor in the body, and it also has to do a lot of other things to be effective as a pharmaceutical, and this is a list here. Chris Lipinski has come up with this Rule of 5 to guide the development of likely successful drug candidates. So not too polar, not too big, not too hydrophobic. It's a really great tool. It's provided a lot of use within the pharmaceutical industry. However, it isn't always successful. And you look at this molecule, the upper left, that's atorvastatin, that's Lipitor. That's a compound that had tremendous patient value and still continues to do so. It also had tremendous shareholder value because Pfizer was selling $10, $12 billion a year, this drug, its peak sales. So, that's obviously a molecule that wouldn't have been very good for that company or the patient population to lose. So there are a lot of places where these predictive tools, you say, how much confidence do we need? How much lack of confidence can we tolerate in our design system? These are really important questions. There's no clear answer to that, and there's likely no clear answer that's going to span a number of industries. Case study, the development of these insecticides. Insecticides that are more broadly acting can cause a lot of problems, and we've seen that, of course. So they can cause electron transport problems, monooxygen, endocrine disrupting activity. So how do you get around this? The way that Roman Haas worked with this group is to target specifically a biochemical process that was unique to insect, to go after something that affected molting. So by doing that, you narrow the range of receptors that could potentially be impacted by this chemical. Now, certainly there's a lot of very important insects out there, so this doesn't completely make all the risks go away, but these are types of strategies that can be applied to try and reduce the risk from using a chemical. Okay, so the key learning points here. So design of preferred chemicals with less hazard holds great promise, and I'd say it's a necessity as well as something that holds great promise. We need to do better. The rate at which new chemicals are being developed really requires us to come up with better predictive tools. And understanding within any chemical industry that the discovery and development process and knowing how to bring these tools in is a really critical task that people who are able to do this well will serve their industry very well, and they'll quite likely do very well within that industry. Being creative and innovative in this area is something that's really important. And once again, the trade-offs are really critical. So just broadly stating, we can't have anything toxic. Well, it's going to be very hard to do that unless we're going to deal with water or something like that. We really need to come up with better ways to affect trade-offs and figure out how to use those, because the world is just not that simple. There's a lot of shades of gray rather than black and white. Okay, questions?
When you were talking about the insecticide,
Mm-hmm.
and the more specific, like targeting molds and stuff.
Yep.
So, is it more expensive when drug resistant with the insects become the ones that are resistant to that specific mode of action? Would it be more expensive to find another drug that?
Well, no, that wasn't a drug. That was an insecticide. That was a bioactive compound that was in the pesticide control area. Resistance, microbes become resistant much more readily than insects. Insects can certainly develop resistance. Resistance is just a huge problem for microbiology and medical microbiology in particular. I'm not much of a subject matter expert when it comes to insect resistance, so I really can't speak to that point very well. Sorry. Any other questions?
I hope that you get a sense of the difficulty that attends the last two talks that we were talking about. One, it's a large amount of information. Secondly, designing molecules. Think about that. In the isosteres that Rich showed, all that was different in many cases was a single molecule or single element in a different location. And if you think about Avogadro's number, and I'm sure you all go to bed every night thinking about 6.023 times 10 to the 23rd, however, that's a tremendous amount of chemical diversity, and we're not exploiting but a very small part of that. And think of it, only one element in a molecule, molecular weight 500 or so, can change whether or not that molecule is a problem. It's an enormous problem. So we're on to an exercise now. So open up your basket and go to Risky Business.